In this video, I want to introduce some key concepts of gene regulation. So here we have different cells. We have blood cells on the left and neural cells on the right. These different cells have very different shapes, as you can see, different sizes, different features, different functions. So they're very different shapes, different functions, different phenotypes. However, if we think about the DNA in these cells, we can ask ourselves, is the DNA content in a skin cell and a neuron from the same person? Are they the DNA the same, or is it different? Or is it mostly the same, or it depends? So just think about that for a moment. So it turns out to be the case that the DNA from different cells of the same person are mostly the same. And in fact, we usually just treat them as being entirely the same. The only reason they would be different is that occasionally there are mistakes, there are mutations that occur during development that can lead to differences. And these can be important in some cases. In particular, these can be important in cancer, as we'll talk about later. But we generally, for the most part, we ignore them. And the reason is that relative to the differences between people, right? So if we're talking about a DNA difference between you and me, or a phenotypic difference, lactase intolerance or whatever it is between you and me, it's probably not going to be due to these mutations. It's probably going to be due to mutations that are in all our cells that I inherited from my parents. So most differences, genetic differences between you and me, are inherited differences, not these rare developmental mutations. And so consequently, when we talk about DNA content for a skin cell and a neuron from the same person, we want to emphasize that they're essentially the same. So what that means is that all this difference that we see, these different phenotypes, is not due to differences in the DNA, so it has to be something else. So what is it? So here what I'm showing on the top and on the bottom is 5 genes. I'm using these things to mean this is one gene, and then this is the same gene, this is another gene, this is the same gene, and so forth. So here in skin and neuron, we have 5 genes, and that would mean that the DNA here is identical. So what I'm using these arrows to indicate would mean that this gene here is on in skin cells, and it's also on in neurons. So that means that this gene is being expressed in skin and being expressed in neurons. But then this gene is being expressed in skin, because we've got a big arrow, but it's not being expressed in neurons. So this gene is off in neurons. And then this one would be on in neurons and highly expressed, but on in skin, but at a lower level, a smaller arrow. And the reason the arrows go right and left is related to the fact that some genes are transcribed in one direction, some in the other, as we discussed in our transcription video. But if that's confusing you, I would ignore that part for now and just think about we have 5 genes here. Okay, so what we're seeing is some genes are expressed in both skin and neurons, some are on in skin, off in neurons, some are on in both cell types, but at different levels, higher in neurons or higher in skin, and then other genes are on in neurons and off in skin. So these are the differences, again, not differences in the DNA, but difference in how that DNA, how the genes are being expressed. And we call this gene regulation, that different genes are on or off in different cells. And so we've already met this in the case of lactase persistence. We said that we all express lactase, or almost all of us express lactase as children, but then some of us turn off our lactase gene in adulthood. Well, that's the same thing, right? That would mean if this was the lactase gene, then we could have a similar diagram, but this would be childhood that the gene is on, and adulthood that the gene is off. So this is the general concept of gene regulation that can be different between stages. It can be different between different kinds of cells. It can be different between different environmental conditions. And so in this video, we're going to learn the first things about how this works. So this is not only important in multicellular organisms, it's also important in unicellular organisms. So Candida albicans is a yeast that has these 2 very different phenotypes. So what this means is that the same cell with the same DNA sequence will sometimes adopt this phenotype with this clear, round phenotype, and sometimes this ovular, opaque phenotype. In Plasmodium, is again a single-celled organism. It goes through a very complex life stage where it's in ourselves and also in mosquitoes. different cell types of the organism are quite different. So you can see here, this is one cell type of the plasmodium. Here's another cell type, and you can see just at a gross morphological level that these are quite different. So these differences, again, are not due to differences in the DNA. They're due to how the DNA is expressed. That is, differences in the regulation of the genes. Okay, and so what's the general way that this works? So in general, products from one gene, so like a protein that's expressed from one gene, will physically bind to some DNA, RNA, or protein from another gene to regulate that gene, sorry, that should be apostrophe, that gene's expression. And so it means we have two things here. We have the regulator, the product that is doing the regulation, and then we have the target, the gene that is being bound to and regulated. So let's look at, let's see what that looks like. So that means we have gene 1. Gene 1 is expressing some protein. And then that protein is coming down and binding to, binding near the gene and influencing its expression. So that means this protein would come down and bind here and influence whether this gene is being expressed or not. So we can have positive regulation. So that would mean when the orange protein is bound, that we get more expression. When it's not bound, we get less expression. Or we can have negative expression. So for a different gene and a different pair of genes, a different blue and a different orange, here what we're seeing is that when the protein binds, that we get less expression than when the protein is not bound. And then another point to make out is that it doesn't have to be 100%, right? Here we see when the orange protein is bound that we're still producing the blue protein just at a lower level. When it's not bound, we produce more. Okay, and so the way we're going to talk about this is gene 1 positively regulates gene 2 at the level of transcription. Okay, so that means gene 1 is expressing a product that is regulating gene 2, either positively or negatively, and what it's doing is it's regulating the degree of expression of gene 2, of transcription of gene 2. So we would say that gene 1 positively regulates gene 2 at the level of transcription. And then we can write that this green arrow just means that it's positive, that gene 1 has a positive regulatory effect on gene 2. Or we could say gene 3 negatively regulates transcription of gene 4, and then we can do that with this block thing, this red block. And so that means that we can get a complex gene regulatory network where we have all these different genes regulating all these other genes. And that means we can contain a huge amount of information that scientists have figured out in this simple diagram. And as we'll learn, this will also allow us to predict, for instance, how this gene will affect the expression of this gene. Okay, so let's go with an example. So blue eyes in humans can actually be due to two different kinds of mutations, either mutations in HERC or mutations in ACA. And the reason is that ACA2 is responsible for producing pigment. It plays an important role in pigment production in the eye. And HERC2 positively regulates ACA2. So that means that we need both genes in order to produce brown pigment, and if either one of them are broken, then we end up with blue eyes. Okay, so in language, we're going to say HERC2 positively regulates ACA2, and it happens to be at the level of transcription. So let's see what that looks like. Okay, so we've got our two alleles, because we're diploid organisms. We got one allele from our mom, one allele from our dad. So we've got two alleles of the HERC2 gene, and we've got two alleles of the ACA2 gene. Again, one from our mom, one from our dad. And then we have a binding site. We can call this the HERC2 binding site, HBS. That's right next to ACA2. Okay, so let's look what happens. If we have functional copies of HERC2 and of ACA2, we get production of the HERC2 protein. It comes and binds ACA2 and positively regulates production of ACA2. So if we have both HERC2 and ACA2, we will produce ACA2, and therefore we will produce protein. And it's worth pointing out, so functionally what matters for production of pigment is ACA2. And so if we want to analyze different genotypes, different mutations, then we want to emphasize whether or not we're producing ACA2 protein. Okay, so let's do some genotypes here. Okay, so some terminology. So we're going to use capital O to mean the functional allele, and capital H to mean the functional allele, and then we'll use lowercase to mean mutants that don't produce protein. So that would mean here is that of our two alleles, one of them is producing protein and the other one is not. So then we can analyze what's going to happen here. Are we going to produce ACA2? Is this individual going to have blue eyes or brown eyes? So if we think about it, these proteins are going to diffuse throughout the nucleus, throughout all around the DNA, and they can come down and bind to both of these alleles. And so we expect to produce ACA2 because we have binding of HERC2, and so we would expect brown eyes in this individual. Okay, what about if we have two mutant alleles? Well, now we're not going to produce any HERC2, so that HERC2 will not bind to the HBS, so we will not get production of ACA2, so we will get blue eyes. Okay, so we could also do some mutants. What happens if we have a mutation in one of our ACA genes? Well, now we're going to have plenty of HERC2, which will come down and bind to the HBSs, but this gene will not produce any protein. However, this one will. It turns out, so these individuals we'd expect to have about half as much of the ACA2 protein. It turns out that that's plenty for brown eyes, and these individuals would have brown eyes. And we can do a double mutant of ACA2. Now there's no gene to produce ACA2, so we'd expect blue eyes. And so another one we could do is, what about if we have a binding site mutation? So this is different. Before we've been talking about mutations in the actual gene itself, the coding sequence. But now we're talking about mutations. We have a perfectly good coding sequence, but we have a mutation in the H binding site. So under these circumstances, we'd expect production of HERC2, but then those proteins would not be able to bind, and so we would not expect production of ACA2, so we'd expect blue eyes. So here's a problem. Do we expect ACA2 production for an individual that is homozygous for a binding site mutation? And so, as I just went through, we would expect no ACA2 production, right? So, so this is our first example of HERC2 and ACA2, a two-gene circuit, we can call it, and some practice on determining what we expect from different genotypes. Okay, so now we can do the rules of regulation, which means basically if we have a complex regulatory diagram, what do we expect to see in terms of the output? Okay, so the first rule is if we have a green arrow, then that means we expect the same. That means if we have an increase in production of product A, we'll get an increase in product B. If we get a decrease in product A, we'll get a decrease in product B. Right? Like if we get more HERC, then we expect that will increase production of ACA. If we have less HERC, that will decrease production of ACA. However, if it's a block, it's the opposite. So now if we get more product C, that product C will come and bind to and repress product D, so we'll see less product D. So that means if we have one of these things, then we're going to see the opposite. When C goes up, D goes down. When C goes down, D goes up. Okay, so that means that if we have a complex logical chain like this, that we can analyze it just step by step. Okay, so we can ask a question like, if A increases, what do we expect to happen to D? Okay, so we can say A goes up. So what happens to B? Well, it's the same, so it should also go up. Okay, now what happens to C? Well, C, if B goes up, that's going to decrease expression of C, so C would go down. Okay, now what about C and D? Well, if C goes down, that means it will be doing less stimulating of D, so D will go down. And that actually answers our question, what will happen to D, but we could continue through to E. If D goes down, E is going to go up. So what that would mean is just by doing it step by step, that we can see if A goes up, then E will go up, D will go down, and so forth. Okay, so what about this one? If H goes up, if H increases, what do we expect to happen to I16? And I'm sorry, somehow this should be a green arrow and a green arrow. So if H goes up, H increases, what do we expect to happen to this product I16? Okay, well, H goes up, so M goes down, so F goes down, F goes down, so Q goes down, Q goes down, so I16 goes up, and so we can answer we expect production of I16 to increase if H increases. So I want to point something out here. We can kind of cheat. So if I give you a long chain of things, actually all we need to know is how many blocks there are, right? So if there's 2 blocks, like here, 1, 2, that means H and I16 are going to go up together. When H goes up, I16 goes up. But if it were an odd, like let's do from H to Q, then it's going to be the opposite. So when H goes up, Q goes down. When H goes down, Q goes up. So that means if we have a complex logical chain, all we need to do is count the number of blocks, the number of red negative regulatory relationships, and that will give us our answer. Okay, so we can do questions like this. If I decreases, what happens to 4? Well, we can just count. If 1 increases, what will happen to product 6? We can just count 1, 2, 3, 4, 5 in between. So 5 is odd, so that means it's going to be opposite. If 1 goes up, 6 will go down. So, and we could work it out. If I decreases, so that means I down, 2 up, 3 down, 4 up, 5 down, 6 up. So if this decreases, this increases. We can just count the 5 because it's odd. They'll be opposite. Okay, so the next thing I want to talk about is autoregulation, which is when a gene produces a product that regulates itself. So far we've just talked about genes that we want to be on or off. So I shouldn't say want, but lactase with the state where it's either produced or it's not produced. But we also want to be able to control the levels in some cases. Sometimes it's not just we want the gene to be all the way on or all the way off, but we want some intermediate level. Right, so that means for this gene, we want to produce some amount of protein, but we don't want to produce a huge amount of protein. So some, but not a huge amount. So one common way to accomplish this is actually to have a binding site for the same protein, a negative regulatory binding site regulates. So this means it's regulating itself, right? So this protein will come over and bind to. If we're producing protein, it will come and bind and repress production of protein. Right, so we can start out with no A. So we have a low level of A. So nothing's binding to the ABS. So that means the gene is not being repressed. So that means that A will be produced. And so we'll produce it. And let's say we overproduce it. We produce more than a moderate amount. Well, then that protein will come and bind to the binding site, and now it will repress it. And so that means over time, as these proteins degrade, we'll get to an intermediate level. So we'll be doing a lot with this kind of stuff, but there's the first introduction. So we call this negative autoregulation. Negative because it's a negative relationship, and auto because auto means self, so self-regulation, so negative self-regulation. So that means it regulates expression. Expression of the gene at an intermediate level. So it's going to lead to expression of the gene at an intermediate level, because if the level goes high, then it will negatively regulate itself and turn down expression. And if the level goes up, it will positively regulate itself and turn up expression. So it's going to lead things to a moderate level. And we can just write this like this. A is negatively regulating A. So here are our main points. I really want to emphasize that regulation is a physical binding of one protein to another. This is not an abstraction. This is not an idea. We're talking about physical interaction between chemical molecules. The second one is the regulator can increase or decrease expression of the regulated gene, the target. And then we can have autoregulation, where a gene regulates itself. And negative autoregulation is a common way to produce moderate levels of gene expression.